This next section talks about T cell activation. So there's a bunch of different types of T cells that exist. The two that we're going to focus on in this class are going to be helper T cells and cytotoxic T cells. And each subset of T cells is activated in a slightly different way. So if we have our helper T cell, a T cell has a T cell receptor. And that T cell receptor is going to recognize and bind to an antigen that is presented on MHC class 2 of an antigen presenting cell. So in step 1, the T cell receptor is going to recognize and bind to antigen that's presented on MHC class 2 of an antigen presenting cell. But that's not enough to get this to activate. We need another thing to bind, and that second receptor is going to go from the T cell directly to MHC class 2, and this is known as CD4. CD for complex of differentiation, and it's number 4. So there's all sorts of different CD complexes, and the different type of CD present on the surface of the cell helps us identify that cell in a clinical setting. So helper T cells have CD4. CD4 binds to MHC class 2. So a helper T cell will get activated when step 1, the T cell receptor recognizes and binds to antigen, which is presented on MHC class 2. Step 2, CD4 of the helper T cell binds to MHC class 2. Once this helper T cell is activated, then it will go help the immune system by stimulating other cells to fight whatever pathogen expressed that antigen in the body. So helper T cells help activate other T cells and other types of cells in the immune system. Cytotoxic T cells are a little bit different. So cytotoxic T cell, which we'll abbreviate TC cell, has a T cell receptor . And that T cell receptor will still bind to antigen. But this time, the antigen is presented by MHC class 1 of an antigen of an infected self cell. Again, this isn't enough to activate that cytotoxic T cell, so we need another arm involved, and that arm is now called CD8. And CD8 binds to MHC class 1. So the steps of activating a cytotoxic T cell are the T cell receptor recognizes and binds to antigen, which is presented on MHC class 1 of an infected self cell. CD8 of the cytotoxic T cell then binds to MHC class 1. Once both of those are activated, cytotoxic T cells are going to kill whatever cell it's attached to. So in this case, the cytotoxic T cell is going to kill the infected self cell. We talked about this earlier, how that's beneficial. So if this is infected, so there's microorganisms hiding inside of the cell, we kill it, and then those microorganisms will escape and we can kill them. We can target them and eliminate them from the body. These infected self cells can regenerate, so they're any type of tissue cell that can regenerate. Unfortunately, there's some microorganisms that can hide inside of our neurons. we don't want to kill off our neurons in order to have access to those microorganisms. So microorganisms that hide inside of our neurons, like the varicella virus, which causes chickenpox and later shingles, or the herpes virus, which causes like human herpes virus type 3 that causes genital herpes, type 2, that causes genital herpes, that is going to live inside of our neurons, and our neurons don't even express MHC class 1. So our cytotoxic T cells will never kill off a neuron. And those pathogens remain unchecked in the body for a really long time. You can have shingles outbreaks monthly for the rest of your life. You can have herpes outbreaks every week for the rest of your life. So that virus in those instances, they stay inside of neurons, which are protected because they do not express MHC class 1. In the case of HIV, so this is different from T cell activation, but HIV involves T cells, so I'm going to talk a little bit about how HIV infects your cells. So we have our helper T cell with our T cell receptor, so our TH cell, and we have our CD4. HIV attacks this CD4 complex and infects our helper T cells. Excuse me. So once HIV gets inside of our helper T cells, it does a couple different things. HIV will go through the process of viral cell infections. So it gets rid of its capsid, it releases its genetic material, and in the case of HIV, it's actually going to incorporate its DNA into the DNA of our helper T cell. So at this point, the only evidence of a virus is the fact that viral DNA is now incorporated into our genome of the helper T cell, into our genetic information, our DNA that's found inside of a helper T cell. When stimulated, this helper T cell is going to start replicating that viral genetic material and start to make more viral copies. Once the viruses start to accumulate inside of the helper T cell, this helper T cell will be killed directly by HIV. But if it's not killed directly, there's another thing that happens here. This helper T cell will start to express MHC class 1 and present an antigen to your immune system. Now in this situation, the helper T cell is acting like an infected self cell. It's saying, hey, come kill me because this is inside of me. Now your cytotoxic T cells, with their T cell receptor, will recognize and bind to the antigen. Their CD8 will bind to the MHC class 1, and your cytotoxic T cells will start to kill off your helper T cells. So what HIV does is not only does it directly kill these helper T cells, but it turns your cytotoxic T cells against your helper T cells. Since helper T cells play such a major role in stimulating cells of the immune system to boost immune responses against pathogens, if you decrease your helper T cells below a certain level, you're considered to have AIDS, or acquired immunodeficiency syndrome. You won't be able to fight infection normally, and you end up dying of subsequent infections that you get because you can't fight them normally. There's also other types of diseases. Like there's a type of cancer called Kaposi sarcoma. So that's skin lesions that form on the surface of the body all over, and that's a type of cancer that can affect AIDS patients. Those nosocomial or hospital-acquired infections are really big with AIDS patients. Luckily today, with the medications that we have, we've decreased viral load in the bloodstream. And we've been able to control viral loads so much that now couples that are infected with HIV, if the man is infected with HIV, he can safely have a baby with a woman, and the viral load in the semen is so low that the woman, his partner, is not likely to get infected anymore. So as long as the patient is taking their medications appropriately, usually you can decrease the viral load to a safe level now. So having HIV today, although there's no cure, is a lot less dangerous than it was years ago before we had all of these medications that exist. Why can't we completely cure HIV? Because the virus incorporates its genetic material into your cell's DNA. Every time that we make new copies of the virus, it's mutating to become a different type of virus that's circulating around. It's difficult to develop a vaccine or a drug that targets HIV when it's constantly changing. And if you know anything about drug development, from the lab bench to actual the end of clinical trials and using it as a medication, it takes on average 10 to 12 years. And lots of money goes into this. So taking that amount of time to develop a drug, by the time the drug hits the market, it's a completely new type of virus that we're dealing with. So there's a lot of complications involved in treating HIV, but for this class you definitely need to know that HIV infects helper T cells through the CD4 arm. And once it gets inside, it not only directly kills helper T cells, but it causes these cytotoxic T cells to turn on your helper T cells, ultimately decreasing helper T cell numbers to a point where you have an immunodeficiency.